Hello, everyone. My name is Natasha Sheybani, and I'll be moderating today's Fireside chat on gene therapy. During this session, we will introduce and discuss gene therapy, as well as some of the considerations that will allow focused ultrasound to revolutionize the impact of gene therapies going forward. As such, it is an absolute honor to welcome a distinguished expert on gene therapy to today's Fireside chat, Dr. Nathalie Cartier of AskBio. Dr. Cartier obtained her medical degree from the Université Paris-Descartes in Paris, where she specialized in pediatrics. She became research director at INSERM and professor at Université Paris-Descartes. Her research interest focuses on the development of gene therapy approaches for severe neurodegenerative diseases. She developed with Patrick Aubourg the pioneer work on hematopoietic stem cell gene therapy for X-linked adrenoleukodystrophy, the first clinical trial using an HIV-derived lentiviral vector. And she's co-PI in gene therapy clinical trials for adrenoleukodystrophy and metachromatic leukodystrophy. Her research lab in INSERM Paris focuses on AAV gene delivery for Alzheimer's disease, Huntington's disease, and spinocerebellar ataxias. Her lab is located in the prestigious Institute for Brain and Spine in Paris and is particularly interested in brain metabolism and in modulation of brain cholesterol. Dr. Cartier was member of the Scientific Committee of INSERM and former president of the European Society for Gene and Cell Therapy. She is currently president of the Scientific Committee for the French Foundation for Rare Diseases. She received from the French government the Medal of the Légion d'Honneur and the Grand Prix of the French Foundation for Medical Research. Dr. Cartier is co-founder of Brain Vectis Therapeutics, which recently became AskBio France, and develops clinical gene therapy programs for neurodegenerative diseases. So on behalf of the Focused Ultrasound Foundation, Dr. Cartier, it is my pleasure to welcome you to this session today.
Thank you very much, Natasha, and thank you very much to the committee for inviting me to discuss with you today. It's a pleasure.
Wonderful. Well, again, it's great to have you. And I'd like to begin with the first question, which is, in a few words, how would you describe gene therapy and its advantages relative to other types of therapy?
Okay, so gene therapy is very different from small molecules. You bring a gene into the body to produce a therapeutic protein, and you do that just one time, and this is going to last for a very long time. So gene therapy is a very important strategy to treat disease, not only monogenic disease, where, of course, one gene is missing, you can replace the gene, but also for multifactorial disease or complex disease, where you can bring a therapeutic protein. And because gene therapy is just a one-time treatment, is a very efficient treatment. And also you can be very precise where you deliver the therapeutic gene. And the last thing is, again, it is a very stable approach. So just one time for a very long delivery.
Wonderful. Well, yeah, I really don't think that can be impressed upon this audience enough, especially with all of the drug delivery efforts that are being highlighted in this symposium. It's really incredible that we could reach a point where a therapy that's only administered once could have such radical impact. So what are examples of brain or other, if you prefer, disorders that you're currently developing promising gene therapy candidates for? And I guess I'll just follow up by asking, if you had to pick one, which do you think is the most ripe for near-term translation?
So I will tell about neurodegenerative disease because this is my field.
Sure.
And with this, of course, I'm very interested, as I said, in monogenic diseases, genetic diseases, but also in complex disease, like you said, Alzheimer or Huntington. And if I have to take one example, actually, I will take two. I will take Huntington disease and I will take Parkinson's disease. Why should I take these examples? Because these are, one is genetic, one is more complex disease, genetic Huntington, the other The Parkinson is a more complex disease, but both affect a special region of the brain, which is called the striatum. and so for which we can deliver the gene locally into the affected region to hope for a clinical benefit. So this is a really straightforward approach with gene therapy.
Wonderful. And as you well know, these are two disease categories for which there has been lots of advancement and innovation on the topic of focused ultrasound. So we'll actually come back to some of that interface in a couple of questions. So let me follow up with another question, which is, what are the limitations or outstanding challenges in the field of gene therapy?
Today, there is two major challenges to go to the patients and to be able to treat a large number of patients. One is the manufacturing of the therapeutic vectors, because gene therapy is based on the use of a viral vector, which is, of course, safe, but can deliver efficiently the gene into the target cell in the body. And the manufacturing of such vector to a clinical grade to do like exactly like a drug is extremely important and difficult, and it costs a lot of money. So this is really an important aspect. The second critical aspect today is to improve our tools for this delivery, and particularly for brain delivery. So this is I will mention two things. One is the capsids of the vector, so the envelope that allows to target efficiently the brain. And one particular aspect, we will come back to that a little later, speaking of the ultrasounds, is to cross the blood-brain barrier. Because this blood-brain barrier in the brain prevents the crossing of the drugs, but also of the gene therapy vectors. So we need to improve our tools to have vectors, to have capsid that can efficiently cross this blood-brain barrier. So this one point. The second point is the specificity. We want to be very specific to be also very safe, not to go where we don't want to go, and go efficiently in where we want to go, in the cells we want to reach. So to that, we have to work on designing the cassettes that we put in the vector. So using promoters, using specific enhancer sequences that can improve the delivery and the specificity.
Most certainly. And that is actually the perfect lead into my next question, which is, based on these challenges, do you believe that focused ultrasound could sort of help to overcome any of the limitations facing gene therapy? And if so, how?
Exactly, because the important thing for gene therapy for neurodegenerative disease today is to simplify the technology and make it more efficient, again, to cross the blood-brain barrier and more efficiently target the diseased cell. And to do so, of course, the best way would be to deliver the gene by simple intravenous delivery, not go by local injection, which is, of course, very invasive for the patients. But intravenous delivery means that we have to bring a lot of vector, some of which will cross the blood-brain barrier, but not very efficiently. The use of focused ultrasound should improve this targeting through the blood-brain barrier in the region we want to target by focused ultrasounds that would allow the vector to cross more efficiently. So this would be a major improvement, yes.
Great. Well, focused ultrasound mediated blood-brain barrier disruption is advancing rapidly in the clinic. So I think there's a lot of opportunity going forward for that exact approach to take place. On the same note, this perhaps was answered by the previous question, but whether it includes BBB opening or is aside from that, I'm wondering which FUS regimes more broadly you are excited about interfacing with gene therapy. Is there anything else outside of barrier opening?
So what we use for opening the blood-brain barrier with focal ultrasound is low intensity ultrasounds that transiently open the barrier based on mechanical oscillation of microbubbles that help the virus to cross this barrier. This is really important because it's a transitory procedure. It's harmful, And so this should really improve the delivery to the patients in a simple manner, so feasible in a large number of places, so limited cost also. So this is really important.
Absolutely. It's critical. And then again, I'm really hopeful that this conversation is prefacing some advanced preclinical, if not clinical efforts that are going to be forthcoming in the very near future.
Absolutely. Yes.
So I'm going to shift gears now and ask another question more broadly about gene vectors, since we talked about this a bit a moment ago. What do you think? We've talked about viral vectors, and of course, these are a really powerful tool for gene therapy, but I'm curious about what you think the outlook is for non-viral gene vectors. As you know, a lot of people in the field of focused ultrasound, and of course, outside of the field, are exploring alternatives to viral vectors. So I'm curious about how you think these formulations compare and whether there are any particular non-viral vectors that you're also excited about.
I think there's a lot of improvement done in that field for the moment. Not so many applications, but I think it's an important field because again, the aim is to be safe, to be easily usable for patients. So in that field, I think the use of liposomes, for example, and exosomes is a really promising technology. Again, the aim of all that is to be more specific in the targeting. So to design all these non-viral particles to more efficiently be addressed to the brain, to the blood-brain barrier, and go exactly where we want to go. It's important because also it might help us to avoid some immune response that we may have with viral vectors, and we're working a lot on limiting, of course, this immune response and improving the specificity. Go to the brain, not going to peripheral tissues, for example. So this is really also an interesting technology, and this could be combined to the use of viral vectors, of course, because you can imagine to combine these particles to improve the delivery to the vector that can easily move to the target cells in the brain.
Absolutely. Well, that's, ultimately it's really fascinating to think about this sort of, dichotomy between viral and non-viral vectors. Of course, viral vectors have taken off so much in the clinic that I'm sort of curious what the pathway is intended to look like for non-viral vectors. So that certainly speaks to the question. Another question that I have now, shifting gears back to FUS, is, we've talked about these really promising approaches in the brain in particular. And of course, with any new therapeutic paradigm, there are technological limitations that need to be addressed. So I wanted to garner your opinion sort of on behalf of the FUS community as to what criteria or unmet needs you would ideally like to see fulfilled by FUS technology for gene therapy applications in the clinic? What would your, ideal focused ultrasound setup or clinical treatment look like?
As I said, my ideal goal is to simplify the delivery to patients. So by combining just one single IV delivery to the use of focused ultrasounds of low intensity, non-invasive, non-toxic, would really hopefully improve the delivery in localized region of the brain for disease, as I said, like Huntington or Parkinson disease, but eventually also in the future, do it more broadly to the whole brain and treat disease like Alzheimer's disease, for example, but also genetic disease that concern the whole brain tissue, and for which it's very difficult to have this diffusion in whole the parenchyma, the brain parenchyma. So really, my goal is to improve the technology so it's harmful to patients. It can be very specific to a region or more broadly to the brain, and with a cost that is affordable to treat a lot of patients. And again, the interest of using ultrasound combined to intravenous delivery would be that we would need much less quantity of viral vector to be administered to the patient. And when we know the cost, as I said before, of the viral vectors so far, we injecting less amount of vector would be a really important issue for the treatment of a large number of patients.
Absolutely. So I think there are a couple points there. I mean, certainly one is that, delivering these viral vectors is already inherently cost effective because of the amount of money you can save by delivering less vector. On that note, I guess for our audience's edification, I'm wondering if you can speak a little bit to what the average cost of a gene therapy is. For someone who comes in the clinic, is that I don't know if that's a reasonable question to answer.
No, it's not a reasonable question because it's a point that is extremely discussed these days. And we have a lot of partners in the field that try to do their best to limit the cost of this gene therapy product by working together, industry, medical doctors, patient association, to make this vector affordable to a large number of patients. So again, the cost is high because the cost of the production and of the research to make this feasible is very high. But because of the improvement we are currently making in the manufacturing, as I said, the cost is decreasing a lot. And also, you have to know that there's a lot of efforts put on making these products available even for very rare disease, so for a very few number of patients through foundations and caritative association. And I think this is a really important point. So again, the cost is high. It can be several $100,000. But at the end, what we're currently doing is trying to decrease this cost, to improve the technology, and to make it affordable to a large number of patients around the world.
Wonderful. And that totally aligns, I think, with the intent of that question. Is really to, impress upon the audience that there are a lot of efforts being made right now to reduce those costs. And that completely parallels the activity in that regard as far as where focused ultrasound is right now as well. So before we depart from this question, I wanted to ask you one more. You mentioned the notion of gene therapy and, of course, in conjunction with focused ultrasound, that this paradigm be cost effective. And so I was curious about what your perspective is on the role of image guidance and where you foresee image guided systems of the ones that you're familiar with helping us in this effort to deliver precise therapies.
So my opinion on that, I'm not a neurosurgeon. I'm not a focused ultrasound specialist. Okay. So, but my opinion is this is extremely helpful to be very precise and to make this first proof of concept that we can be beneficial to patients with this technology. This is critical as a start. But to my opinion, in the future, what we need to go to is as much as possible a simplified technology. So it could be feasible in a large number of places for a large number of patients. So hopefully this technology will move towards more simple ways, like using simple neuronavigation systems, or also so it's more easily doable in different places around the world.
Certainly, and it's actually very exciting and timely that you bring that up, because I think some of the content highlighted in this symposium actually speaks to how much advancement is happening already on that front. Yeah. So again, very timely. And that concludes a lot of the scientific or technical questions I had for you. But if you would indulge one more, I simply want to ask this for all of the trainees out there who are, looking at your record and are in deep admiration of all your accomplishments. I want to ask, what brought you into the field of gene therapy? What inspired you to do this work?
Look, I'm MD. I was grown in the genetic and biochemistry field because my father was a biochemist and an MD also. So I was born in all that, and I loved genetics, and I thought being able to use gene engineering was a fantastic tool. Initially, I did transgenic mice, but I felt this was not what was really important to me. I wanted to use gene manipulation, gene transfer to treat disease, not only to make disease models in animals. So I focused on gene therapy as soon as there were vectors that could be usable for gene therapy. And you mentioned that I did some work with a lentivector to start with an adrenoleukodystrophy. And this, for example, was a very important step, the discovery of such vector that could be used to treat patients, And for me, it was incredible to think that we could be beneficial to patients with such technology. So my goal is to do that forever.
That is deeply inspiring. I think for anyone who's listening to this, it's really wonderful to hear that aspect of your story. And at the expense of sounding very cheesy, I suppose it was in your DNA, right? The idea of pursuing genetic engineering and gene therapy. So, that concludes my questions. If there are not any parting thoughts, I just want to reiterate that this has been a very fruitful discussion. Again, hopefully it marks only the beginning of many more vibrant dialogues to come within the FUS community about ways to advance gene therapy. And Dr. Cartier, I want to thank you so much again for your time and your invaluable insights today. And I believe this concludes our session.
Thank you very much, Natasha. And it was a pleasure discussing with you today. Thanks.
Thank you.